So hello everybody and welcome to NRGene's webinar series. Today the webinar is going to be focusing on cannabis, specifically on how to leverage genomics to accelerate your cannabis breeding program. We'll save time for questions at the end and we encourage you to ask them. So you have two options. Either you type in your questions during the presentation using the questions tab on your the left of your screen, or you can use the hand icon to signal us that you have a question. Then we basically give you the right to speak and we can have a conversation. That will happen at the end of the webinar, as I mentioned. This webinar is also recorded and will be posted on our website. So with us today, we have Dr. Uri Weishaus. Uri holds a PhD in plant and microbial genetics from the Hebrew University in Jerusalem. And his academic background is in genetics of plant secondary metabolism pathways. He's been with NRGene since 2011 and spent most of his time in the company focusing on cannabis genetics. Uri, the stage is yours.
Thank you. So hello everyone, joining from all over, I guess mostly the US and Canada. I'd like to talk to you guys today about cannabis and hemp breeding and what can you expect from this talk. First of all, addressing the challenges of breeding cannabis and applying genomic tools to this fairly new field. The first steps in developing new varieties and breeding programs. And NRGene's unique solution to accelerate breeding with your specific germplasm. And we are, I have intended this talk to address audience with varying levels of technical expertise in genomics. So we do welcome deeper technical questions in the Q&A part following. Okay. So just a bit about NRGene and what we do. It's important for us to mention that we are not a cannabis company. We are a big data processing, data analysis company, working mostly with companies in all other crops. Cannabis is another crop to us. We do not hold germplasm or develop our own breeding program or our own varieties. We've been around since 2010 with over 40 employees, mostly in Israel, with the big R&D center. Over 25 people, mostly PhDs in our R&D team, placed in Israel, with offices also in San Diego, and over 100 customers, mostly seed business and academics. In the cannabis space and hemp specifically, we have several clients, big and small, in North America, with also clients in Switzerland and Israel. Okay, so just a bit of reminder about the plant, Cannabis sativa, one species, very arbitrary definition of hemp and marijuana or cannabis type, drug type. This was a man-made decision, legal decision to discriminate between the two, but this is one plant. They cross, they pollinate, they transfer genes from one another. Usually the hemp type is characterized by 0.3% THC, the psychoactive ingredient, whereas anything above that is called marijuana or drug type cannabis. Specifically, you could also discriminate between the two by the uses. Marijuana type is usually for oil or secondary metabolite extractions with a varying degree of THC above 0.3%, whereas the hemp type is usually for other applications, seed, fiber, in recent years, CBD oil, which is the second most important component of cannabinoid family. Which brings us to the point where most breeding programs are, whether you have a breeding program that has started or you plan to initiate ones. Most of the companies that are in the seed business or in the cannabis growing and breeding business have this kind of mystery envelope. It's a treasure. How do you make an inventory of this treasure? There have been various sources of obtaining seeds, either your heirloom collection, legacy strains, purchase seed, siblings of good varieties that you have, but you can't keep them forever at veg state, so you keep them as seeds, but you lose the information about them. And then you have spontaneous and generated mutations, crosses. That says Purple Haze 164, and the 64 is crossed, and you're not really sure what's in it, but you won't throw it away because there's a lot of potential in there. And then there's night breeding and other various dubious ways of seeds that have gotten into your collection. They're all part of the collection, and most companies are stuck on where to start. Another aspect and trouble of trying to start or proceed with a breeding program is the naming of varieties. Currently, the names mean absolutely nothing. Anybody can name a variety with any name, and it could mean different things. There have been attempts, for instance, by Phylogenetics to create a database connecting genetic information with names. For instance, if you deposit a sample that says sour diesel, you get a star in the galaxy, which means you're close to other sour diesels, and that's about it. You may have similar profile on the metabolites, but maybe the overall genetic distance is pretty close, but your variety will be completely different than the other sour diesels in there. So it has very limited use for breeding. And the real reason, I guess, why all of you have joined the webinar today is this graph. This is actually from June of last summer. There are more updated versions of this dating to last August. This is from Health Canada, and it shows the supply versus demand. And as you can see at the bottom is the demand or the sales of cannabis versus dried or unprocessed and processed surplus cannabis. So this data is for Canada, but it also may relate to various states in the US where there's surplus cannabis either for CBD or for other uses. And it doesn't take much to see that if demand is way down here and supply is up here, prices will go down. And if you're a cannabis producer or grower, there is prices will go down, so you have to be either very, very efficient to compete or have unique varieties that other players do not have. So what's really keeping cannabis from becoming a standard crop? Okay, so there are two categories that are preventing cannabis breeding and cannabis to become standard crop in which ordinary breeding takes place. The first one on the market segment, I guess, would be a very long time to market. There's not much we can do about that. This is the biology. It takes a while to make and generate a new variety. With our technology, that time is shorter, much shorter, but it's still a lot of time. And then the return of investment will happen only in 3 or more years. So that's a limiting step. Next factor is very expensive phenotyping if you're looking into chemotypes, and the seed collections, as shown earlier, are very large. So there's too many plants to handle. Usually there's a limiting space, so you can't grow 5,000 plants and phenotype all of those. And then again, there's difficulty in protecting your IP, and it's very easy to steal your clones. Any disgruntled employee can walk away with a stem, and your hard work is stolen by somebody else. But that situation is gradually changing with PVP laws improving and variety protection and registration of varieties in database. Also, the recreational or the medical market are used to working with clones. So if we're trying to move towards seed production, this will require infrastructure or other handling of different handling techniques towards cannabis. And then one of the other major constraints is that breeding and production compete on indoor space. and you cannot have breeding and production in the same area, mostly for the risk of pollination and licensing issues. And then there's the biological aspects. There are no seed banks, and you cannot, it's very difficult to import germplasm collections from other countries. There is difficulty in keeping males in the facility because crops would be pollinated, and you need very strict control over what gets pollinated by what other plants. Stable polyploids and other shortcuts that make breeding more efficient are rare in the cannabis industry. The plant is dioecious or monoecious or hermaphrodite, so this makes the breeding difficult. You have to be able to convert males into females and vice versa to create successful crosses and inbreeding. There are no inbred lines. Cannabis has very strong inbreeding depressions, so it's hard to find true inbred lines. I'll get to that later. And there are no double haploid shortcuts, at least not yet published for the public domain to use. So when I relate to breeding, breeding means many things to many people. What we focus on and what the market mostly wants is at this point is conventional breeding, where you're taking your elite variety, crossing it with a donor variety that has a different unique trait, do some back crossing, meaning repeated crosses on one of the parents or elite parent, till you end up with elite variety with desirable traits. Obviously a very long process consuming a lot of space and cost for phenotyping and keeping that program running. The other types of breeding are either genetic modification or genome editing. They're both shortcuts to doing conventional breeding. I won't go into the details of that. I'll just say that genetic modification requires transgenic, introducing genes from other species. And genome editing is a fairly new technology that hadn't been so yet used in cannabis, mostly because of tissue regeneration issues. And I do think that right now the cannabis industry and the crowd is not ready for GMO in cannabis. Okay, so why even perform molecular mapping? The main reason to do this would be, first of all, to find a trait and then to use the markers for generating the desirable traits in your germplasm. Specifically, this is an example from southern leaf blight in corn, where you find a trait, it is mapped to a specific gene, and you can now move that gene not by genetic engineering, but standard crossing and markers into your desirable strain. Obviously, this is a nice story here, but most traits are not single genes. You need to transfer either quantitative traits, meaning multiple loci in the genome have to be transferred. And usually the breeding targets are multiple traits for one line. So you need to look at the entire genome and make sure entire chunks of the genome are present in your elite germplasm. The third reason to do mapping is research and isolation of gene targets, either for gene editing or transgenes. As I said earlier, the cannabis industry is not quite ready for this or not quite accepting this yet, but the pharma industry will welcome genes and new IP for synthesis of new drugs or candidates. So the discovery and identification of candidate genes is also a very important target for companies. We rely our genetic efforts on the presence of genomes. Why do we even need a reference genome? And NRGene starts with a good quality reference genome. One of our best strengths is the extraordinary assembly quality of our reference genomes. This because, as this graph shows, in rice, for example, since the year 2000, when the first rice genome was published, about 50% of yield-related traits were discovered after the rice genome was present. And it's a significant driver in trait discovery to have a genome. When you compare that to the lower graph in blue of wheat that did not have a good reference genome or hardly a reference genome at all until last year. And as you can see, there was hardly any increase. Almost 4 traits were cloned for wheat breeding. Until we came up with a collaboration with several research groups, and we've published the hexaploid bread wheat genome, which is a very hard, very long and complex genome. We've done since that over 11 wheat genomes, very challenging task, but we've ended up with a very, very high quality assembly. And since then, over 100 traits have been identified, and we expect the graph to be exponential, same as it was in rice, for traits related to wheat breeding. So a little bit of terminology for those of us who are not breeders. When I say an inbred line or homozygous line versus an F1 hybrid, homozygous lines and inbred is a line or a variety of a plant that has been made by repeated crosses, self-crosses, to have 2 copies of the chromosome identical. So if you have 2 parents like B73 and Missouri 17 here, the 2 copies of the chromosome are identical, and the progeny of that cross will create a true F1 hybrid, which is 1 copy from the female and 1 copy from the male parent, and they're all identical. So you can keep producing those seeds, and they'll produce identical clones or identical, plants in the field. Also exploiting a phenomenon called heterosis or hybrid vigor, in which the progeny of such cross is superior to both the parents. In cannabis, as a different species, the situation is quite different. We do not have that many inbred or fully inbred lines, or at least not yet. So what we do have are unstable or semi-stable seeds. These are obtained by crossing 2 clones or 2 heterozygous parents. So maybe some of the genome has 2 copies that are identical, or some regions of the genome are identical, but some are different. And when you cross and produce seeds, you get the identification of the traits. Some of them may be similar, but some of them will segregate. So you get different performance in the fields, different plants. And this is what currently most of the hemp seeds sold. This is the situation with most hemp seeds. In order to produce true hemp seeds, you need to generate stable inbred lines, as in this illustration. You take female and male, cross them, and get all identical seeds that will perform identical in the greenhouse or in the field. And currently the industry works mostly, especially in the cannabis side, not hemp, works with clones, where you take heterozygous, not identical copies in each chromosome, and reproduce those vegetatively and plant them as is. So when you're breeding for cannabis, the market for breeding and the targets is very, very segregated, meaning there are many market segments depending on your breeding goals. So the end point of what you want to breed for dictates how you breed. For example, if you want to breed for CBD content, higher CBD, and you need flowers, and you do not want seeds in there, you cannot in the same population breed for high oil content in the seed, obviously. If you're going for fiber and biomass, you're not necessarily breeding for other traits. And if you're breeding for plants in the field, it's very different than breeding for plants in the greenhouse. As an example of that, there are many agricultural traits that haven't been touched in this species. For example, the pruning, most of the agricultural important crops have been dealt with genetically to select for the right plant archetype. In cannabis, this isn't the case. We did not have that information. So for instance, plant branching, this could be a trait controlled by single or 2 genes, master genes that control branching, and having control over branching would allow to cut costs of pruning that are currently maybe up to 10% of the growing costs to shape the plants like you want them to. And we know, for example, that in teosinte in maize, teosinte is the wild progenitor of maize. Only a single gene, TB1, for instance, exists that makes the shape from the bushy teosinte into the single stalk maize that we know from crops. And if we could find this version of the gene in cannabis, we may have good control over branching. We know, for example, in cannabis that there are different plant architecture by the classic or old classification, Indica, Sativa, and Ruderalis. There are the same clone, the same species, but there's different morphologies and different features to each group. From the genetic standpoint, it means that there are multiple alleles present in the species that we can take and use in breeding to achieve different plant architectures. If we move up to more complex traits, and I've been asked many times, can you breed for yield? So obviously yield is not a trait on its own, it's a mix of multiple traits. So if you breed for yield and you desire higher cannabinoid yield, you need to break down the specific components of yield into individual traits, what we call compound traits. For example, trichome density, we breed for that, or the number of buds or the size of buds, we breed for that separately and then combine multiple traits. Now, do you need genomics for breeding? No. This is kind of a surprise coming from a genomics company, but cultivars have been generated without using genomics, using classical selections, and this is an example from Clark and Maryland 2017. The generation of skunk number 1, a high quality variety, cultivar recreational, I would say. There's a nice breeding scheme and an effective selection program. As you can see, starting from year 1 to year 11, this required over a decade. Cannabis industry does not have this time. We need shortcuts. This is where NRGene comes in, and our shortcut is a new approach to breeding directly with the heterozygous germplasm, your clones as starting material, with no need to stabilize parents first, or no need to generate recombinant inbred lines or other starting populations for the breeding. So as an overview, we take large-scale breeding populations, we analyze the DNA of individual plants, sequence that in standard sequencing procedures, but we do have our unique protocol for sequencing. Then we do data analytics, and we provide information markers to facilitate breeding decisions, which plant to cross, which plant to keep, and which plants to discard, eventually leading to new inbred parents or semi-stable clones in the process. So you aim for seed, but you could find a clone on the initial steps of the project and propagate that and keep selling that to kind of cut down on costs with the breeding program. This part, our analysis from DNA to actual breeding decisions, takes us about 9 to 12 months, depending on how fast you deliver DNA from the progeny. I won't go into the details of how the technology works. I'll just present these oversimplified diagrams, cartoons here, because this is a lot to take in for people who are not bioinformaticians. So in an overview, what we do is we have good reference assemblies of one or more of the parents in a population. We assemble the de novo of a genome, whether it's a homozygous or somewhat homozygous or a completely heterozygous clone. We assemble those, we analyze those, we compare multiple genomes and create a pangenome. Which is a theoretical comparison. We can overlay on top of that a lot of other information. So we can detect all the structural information, deletions, inversions, and so on. And we could also overlay gene content and say this gene is present in one genome and not in the other. Once we have this type of data organization, we could look at haplotype blocks, which are depicted here in different colors. And For example, we could say which one of these haplotype blocks that are inherited as a block, and we can generate specific sequences, markers, that are unique to each one. So if we want the green line or the green haplotype, we can select for this marker. We could also look at the information in this insertion, and it will only enable us to only select this haplotype of all the group. Once we've done that, we could look at populations, zoom into association studies, examine breeding populations, progeny, and discover gene content associated with traits, QTL mapping, and also generate markers for all the applications. Another feature that is unique to NRGene is the phasing of the genome. Because we do our sequence difference and our assembly in a different way, we phase our genomes, meaning we can separate the maternal and paternal haplotypes. Each of your clone varieties contain 2 chromosomes that are not identical. A traditional collapsed assembly would take and assemble one line of DNA sequence, and in cases where there's difference between the male and female, the algorithm usually selects one of the 2 options or merges them into something called chimera, which is a wrong assembly, and you get sequences that are not truly there. Our assembly separates the 2, so you basically assemble 2 genomes for each plant, and you know which ones came from the female and male parent. Specifically, it also allows us to really organize genomic information. We could look at huge databases, and this is example that we looked at information coming from the public database. We've also done this for client genomes for clients we cannot show, but this is information with public genomes. So as you can see, there's been a huge disorganization, or the naming of chromosomes is very different. We could actually align, we could choose one of the reference genomes, for instance, CBDRX from Sunrise Genetics as the reference point, and then map all the other genomes based on that, and determine that, for instance, in the CBDRX, chromosome 8 is equivalent to chromosome 7 or 5 in other assemblies, whether done by us or by others. Another demonstration of what we could do with this is find candidate genes. We've found a candidate genes for autoflower only based on copy number variation and some phenotype information that we have. So for instance, we've found a gene that is duplicated with 100% conserved in the CBDRX, or one of the genomes that we looked at. Whereas the autoflower varieties that we looked at, such as Finola, and one of the assemblies we did, we found a gene duplication that exists in some of the varieties and not in others. This entire cassette as a whole was duplicated, and it's highly conserved at the protein level to 100%. So we think this is a very important candidate controlling autoflower. Just an example, and this is just by our ability to compare multiple genomes at high resolution. We've also done this type of analysis on gene content for cannabinoid synthesis pathway. We can say that there is huge diversity in gene content and gene duplications for various genes in the cannabinoid pathway. And now we can look at interesting varieties and say, oh, there's a missing gene here. What does it mean? Can I use this in breeding? So the most useful application that all our technology culminates to, and the highly applicable service that we offer for breeding is really phase haplotype markers. This is a highly applicable tool for breeding. We take your germplasm, 2 heterozygous parent clones. You generate the cross. We cannot touch the plants, so we need as input heterozygous clones. We analyze the parents, the 2 of them, and then we also take phenotype data, trait scoring from our clients. We associate in the F1 population or F2 population, we analyze those at low coverage and lower cost, and we associate the phenotype of the parents or of the population with specific regions in the chromosome. For instance, if you want to have a specific phenotype, you have to have the dark blue phenotype in the in your clone or seed. And we decide on being homozygous or heterozygous. We can do all of that. And we generate markers that enable us to pinpoint this particular region and a phase genetic map used in the breeding process. Once we have that, there are repeated cycles of marker-assisted selection, much efficient than random crosses and selections. We provide the genetic markers. You then transfer them into PCR assay, very cheap and robust screens. You look for the markers and decide which plants you want to keep and which plants to discard, eventually leading to either stable inbred parents or mostly inbred parents as for seed production, or a semi-stable clone either for as a partial step or interim step or your final product. All of that could be used and verified and then also used for IP protection. Going back to the mystery seed envelope, so now that we have a set of tools to breed, where do we start? So you still have huge numbers of plants to screen for, and you need to start at founder populations. So what we're offering for this is to have plants grown to seeds, and then analyze very cheaply, very low quantity and coarse analysis, which is kind of a diversity analysis. It's an inventory assessment. So we create this type of phylogenetic tree based on genetic distances and phenotype information. So for this example relates to flowering time and CBD content. So we can divide based on phenotypic information and genetic distances, all the 5,000 samples or 500 samples, and then select individuals from each clade and use those as founder parents and avoid the redundancy and need for multiple phenotyping. So once we've taken that, we move to selection of founder lines, either in a nested population. Minimal population would be two parents, probably more cost effective to use one female and two males, but we could go from there and duplicate this structure many times and work on large populations. So we select the parents based on this, and then we move to generating populations of 200 plants each, a very low number of plants to analyze. And in these populations, we map up to 5 traits in a population and generate markers for those. So this is the final example of how the entire process looks like. For flowering time, you take two parents that are very different in the flowering time, you cross them, you provide phenotypes, which are flowering times. We do our association and marker association and trait mapping, provide genetic markers. And these genetic markers are there then used for the breeding. And we could also explore the regions of interest for candidate genes and downstream applications with that. And I'll end with saying that there are multiple applicable traits to do this. Anything you can provide us phenotypes of, anything you can measure accurately and has segregation on F1 or F2 populations, if you can measure it, we can map it. So to sum up this talk, I would say that cannabis presents a unique breeding challenge, both on biological and the operational aspects. NRGene offers solutions that simplify the new variety development, trait marker identification, and gene discovery. We have addressed by these solutions most of the breeding challenges and accelerate the breeding, adding 3 major advantages. One of them is a low number of plants, so smaller space required and obviously lower cost. Fast analysis, starting with genetic material that's inbred, that's hybrid, or minimal need for phenotyping, only the small population used for the marker generation is phenotyped. After than that, you don't need look don't need to look at phenotypes anymore. You look at genetic markers. And then we're generating all of this specific to your germplasm, so you don't rely on other people's data and proprietary assets. And of course, NRGene is here to provide assistance in all your breeding needs. We're happy to answer all questions on cannabis breeding. I know this presentation had been maybe a little technical for some and maybe simplified for others. We're welcoming people to send us questions, either deep technical questions or even beginners questions. And with that, I'll take questions.
All right, so I see some questions here in the audience. We'll start with Larissa Craig. I'm sorry if I'm butchering your name. So I'll give you the right to speak. Right, I think you're self-muted, Clarissa, so if you can unmute.
Can you hear me?
Yes.
Thank you. I wanted to thank you guys for bringing this information forward. It's definitely something very complicated for us non-scientists, but you guys did a very good job at doing that. So thank you.
Thank you.
Yeah. So my question is, so I'm a patient myself, and I work for the National Cannabis Industry Association. So we love seeing new science and ways to improve the regulated market. My question is there, as was stated here, there seems to be somewhat of a pushback from cultivators in the space about GMOs and genetically modifying, of course, the plant, but at the same time, this is what people have done. Even before cannabis was illegal, all of this time was to perfect genetics, but sometimes without the access to scientists like yourself. So recognizing that we do need more science to perfect genetics of cannabis plants to better serve people, what are some sort of pros and cons, I guess I could say, of mapping and measuring these markers in terms of, say, the preservation of land races and other aspects of genetics?
Okay, so land races have been around, and they're usually kept as seeds or preserved as clones, and they have huge diversity in them. Cannabis is a species that has hyper diversity. It's ultra diverse, and it's very important to preserve these because they are a source of traits. For example, if you have varieties that have been domesticated and cultivated and used in the cannabis industry, there's always a need for new sources of genetic traits, for example, disease resistance. So the preservation of land races is very important. So as I mentioned, in the seed banks that people have, their collections, usually people also preserve land races or heirloom strains obtained from various parts of the world or seed banks. Very important to preserve, very important to analyze, see the phenotypic and genotypic diversity as sources for trait breeding.
All right. Thank you, Clarissa. We have some more written questions, some interesting ones, Rory. First of all, we have Venkata Buri asking, is there a demand for insect resistance? And what kind of rare common pests we're talking about?
Okay, so this is a geography question. There are multiple pests and pathogens that attack cannabis depending on where you grow and how you grow. If you're indoor in Canada and it's I don't know, -10 outside, not that many insects. And if you keep a good practice in your grow house, not that many pests. If you grow outdoor hemp in Colorado or other places, obviously you have everything come to your fields. So there are major pests that disturb cannabis and hemp grow. This is very, very specific to geography. Just to name a few, there's some pests that are not there's a new aphid that likes cannabis. There are some borers, stem borers that attack cannabis, spider mites, but most of what people are concerned with are actually fungal pathogens. Powdery mildew and botrytis are by far the greatest enemy to growing cannabis and hemp.
Right. Thank you very much. The next question is, what is diploidy level in cannabis?
So standard cannabis is diploid. There have been reports and people have said that they've generated cannabis. Obviously, the holy grail would be to generate triploid seedless cannabis. I haven't seen any reports that have done that. There is the cannabis genome in some varieties is not very stable. So I've seen reports and actually seen some plants that are tetraploid, but a lot of times they tend to revert back to being diploid or some weird non-stable inheritance of the chromosomes. So it's not very easy to generate polyploid cannabis, but I won't say it won't be done. You know, it's as more and more people go into this industry and start breeding and play around with polyploidy, we will have more of polyploid cannabis.
Thank you. So we'll take a couple more questions. We have Seth Crawford asking, can you describe how your haplotype is to WGA's high-resolution sequencing like PacBio SQL?
Okay, so this is a little bit of, if I understood the question correctly, this is comparing a sequencing technology to the outcome. So, for instance, PacBio produces long reads and a fairly high error rate that needs what they call polishing or generating short reads to correct these errors. So our technology uses standard Illumina reads, different combination of libraries. We use anywhere from 6 to 8 types of short reads or synthetic long reads, and 10x genomics artificial long reads. We use that. We create a lot of redundant information. Later a slide that actually specifies exactly how it's done. But the redundant information allows us to handle highly repetitive and regions, very complex ones, including the CBD and THC cassette regions. Those are flanked by highly repetitive regions, and we do a very good job at resolving those, and therefore we can find more copies of candidate genes in this pathway. Cannabis is very, very rich in highly repetitive elements. Our phasing technology uses the fact that we make a lot of redundant information, a lot of overlapping pieces, and therefore we could assign specific scaffolds into different we phase our genomes, meaning we know which ones come from which parent.
All right, thanks. Okay, so Ted Klein is asking, do you see phenotyping as a bottleneck, especially for small operations and growers that might be interested in breeding?
Yes, definitely yes, mainly because of cost. If you're phenotyping several traits that are cheap to phenotype, for instance, flowering time or plant height, then this is not a bottleneck. You send somebody in with a ruler or a calendar and take down the flowering times. If you're trying to look at cannabinoids, people usually look at, I don't know, the 9 most dominant cannabinoids or 5 most dominant. That begins to be expensive. If you're looking at rare cannabinoids, you're interested in CBL or something even more exotic. Then we're looking at HPLC and advanced lab phenotyping or chemotyping. We worked with a company that looks at 150 cannabinoids. So obviously this is extremely expensive when you're trying to do this on large scales. One of the advantages of what we do is that you need to phenotype only 200 individuals. And then you look at genetic markers that are very cheap, and there's no more a need for phenotyping after that.
All right, thank you, Uri. So unfortunately, we can't answer all the questions live. There are many, but rest assured that we're gonna be answering all of them in writing. You're gonna be getting the answers to your mailbox. Uri, thanks so much for taking the time to share from your expertise. If you've, and you guys, if you've missed any part of this webinar, we will be sending the recording over by mail. And I also encourage you to continue the discussion with us. We'll be very happy to do so. Also, this webinar recording will be posted on our website. I also welcome you to visit our website for further information and write us to this email address, info@nrgene.com. So to sum up, thank you for joining today and stay tuned for our next webinars. All the best.